In this module, we will discuss the homologous recombination and the transposition, the mechanism how the cell continues and how the cell perform this duty during the division or during the mitosis. HR has common feature in all cells with some variation. This mechanism is entirely and extremely important to maintain the characteristics of the progeny of the cell there. Now, homologous recombination is the exchange of DNA strands of similar or identical nucleotide sequences. In the chromosomes where the DNA strands are there which maintain the characteristics of the cell, they are present, and those strands, the similar strand which has the information for the properties of the cell, that is the identical nucleotide, there is an exchange of DNA. Now, it can also be used to direct error-free repair mechanism of the double-stranded DNA. This is the basic characteristic. This is the basic need of the homologous recombination in the cell when they divide. As you can see in this picture, that they are different sister chromatid and the homologous chromatid. When they join together, they exchange the chromosomal segment, and the new DNA strands, they are originated. And remember that they are not so varied or they are not so different from each other, but they keep up the information and they keep up the progeny of the next generation. Now, approximately 10 to 30% of fertilized human eggs are aneuploid. Now, what is the aneuploid? Aneuploid, they contain an abnormal number of chromosomes. You see, in the chromosomes or in the homologous recombination, there are chances that the sum of the abnormalities, they are produced. And during this process, those cells which has got the abnormal chromosomes, they are produced, and abnormal produced that are actually abnormal chromosomes, they make the cell to die. Now, leading cause of aneuploidy is the reduction in the number of abnormal placement of the recombination event during the meiosis. Remember, we are still studying the different stages of the meiosis and how cell maintains its progeny, how cell maintains its consistency of the next generation, so that the DNA which is damaged, this is repaired automatically. Recombination repair is, as a matter of fact, plays an important role in allowing cells to deal with DNA damage by so-called recombination repair. Whenever there is a production or there is a division and the chromosomes, they divide, the chances are that the DNA damage occurs. And this DNA damage is prevented by the recombination repair of the cell. Biological roles of recombination are, they are generating new genes, all allele combination, that is the crossing over during meiosis, generating new genes from immunoglobulin rearrangement, and integration of a specific DNA element for the viruses, and last is DNA repair, which is the important and the significant contribution of this recombination. Types of recombinations are: one is homologous recombination that occurs between sequences that are near identical. That is, on the DNA, the sequences which are just similar during the meiosis, it occurs between that. Then that is site-specific. Site-specific occurs between sequences with a limited stretch of similarity, and it involves specific site for specific genes. And the transposition of the DNA element moves from one site to another. Usually, little sequence similarity is involved. So DNA element, which is responsible for some characteristic, it moves from one site to another on the chromosome, and this is the transposition recombination. So there are three types of the recombination which we have studied. As we can see here, that there is a single place or single exchange on the top. Then there is a double crossover, that where there are two sites are involved and the genes crossover. Then there is an intramolecular crossover, that is the DNA. It bends and certain specific sites, they are moved and the new segment is produced. And then there is another intraposition where the DNA, the two strand or one strand, it bends, makes a loop, crossover is there, and a new segment or the new DNA which is produced over there. Now, what are the practical uses of this recombination? It is actually used to map the genes on chromosomes. That is, to map the gene that where that gene exists on the chromosome. And the second one is it making the transgenic cells and the organisms where the new specific particular cells which are generated, which are entirely different from the new, or they are the cells which have some disease immunity, which have some resistance to some viruses, and like these things.